told me, let's start a project and search for H1 in yeast. It would take you approximately several months, and it would be good to make a PhD thesis. Fine. It was right. She was right. We started, and the timing was not right. Instead of several months, it took me 12 years. Yeah. Fine. But it was very hard to extract H1, I mean the protein H1 from the yeast. And then the genome was not sequenced yet of yeast. So it was very hard to extract it. And because of this, instead of extracting, I just express an exogenous histone H1 from sea urchin in yeast cells. And what happened was this picture, which showed apoptotic-like process in the yeast. And then we published the paper, but we were very curious to say apoptosis in yeast. Nobody showed some pictures, such pictures. We were very careful, but now we can go to conferences about the apoptosis in yeast. It's easy to see. So the H1 from sea urchin is killing the cells by some kind of apoptosis. And let's take the time machine and come back in nowadays. No, the movie is not starting yet. Let me try again. Oh yeah. So this is a nice movie about how we know, what we know today about the organization of eukaryotic nucleus. We know that chromosomes are taking their place, like chromosome territories and special places in the nucleus. But important for me, for my talk, is that they are making loops with certain sizes. Okay. And because of this, I wanted to see whether I can develop a method to see these loops easily instead of high C, etc. So I turned to the method called comet assay, which is shown here, and I will not go just now in details. But if you treat cells with the genotoxins and put them in an electric field, you obtain comet-like images. And the good thing is you can measure the size of comets, the intensity of the nucleus, intensity of the hull of the comet, and of course intensity of the tail. And therefore, you can make different kinds of equations and make this very beautiful in graphics. Okay. And then I decided, instead of using genotoxins, to use just enzymes which are very, very convenient for chromatin structuring. And after that, after putting the, making several cuts in the nucleus and putting the cells under the electric field, I obtained the same picture of comet. Well, the real picture is like this. Then we, with my colleagues, realized that actually we are obtaining different shapes of the comets. This is not a picture from one experiment. All these comets, this is a mixture from different experiments. I'm just showing it to show that there are different shapes of comets, and they are obtained by different enzymes or genotoxins. And because of these shapes, we decided to measure and make something more using this method. And by measuring approximately 18 different parameters, we designed a software program. This software program could give you different differentiation between types of DNA cuttings, whether they are single-stranded or double-stranded, and you can differentiate apoptosis from necrosis, and you can even guess what kind of genotoxic agent is used to make cuttings in DNA. So by using all our knowledge, we decided to measure chromatin loops in the nucleus. And it's turned out that by measuring, we show that general loops in the human cell nucleus is approximately 1 megabase. And what about yeast cells? It's 0.2 megabase. It is strange because, as we know, yeast are approximately 100 times smaller than human cells, and they've got 100 times less genomic DNA. But the loops are almost the same size. So we can tell that there is some kind of evolutionary conservation of these loops, I mean general loops in the nucleus organization. What about the yeast without linker histones? Because people found the gene about linker histone, and it is easy to delete this gene, and the cells are viable. Strange, but they are viable. And as you see, the loop size of cells without linker histone is a little bit bigger. Not so much. The strange thing is that cells are viable. Usually people think that if you remove H1 from the DNA, from the chromatin, you obtain some kind of differences in the nucleosome repeat. However, you can see this picture. There is no difference in the nucleosome repeat. But then we looked more carefully, and we saw that actually the chromatin of the mutant, knockout mutant, is digested faster than in the chromatin of wild type. And you can see here some kind of pausing in digestion. It's repeated in several experiments, and it is approximately 4 nucleosomes. Around 4 nucleosomes, you've got a pausing of digestion by micrococcal. So the meaning this is probably because H1 in the yeast are in less quantity than in the human cells, and probably it's bound for every 4 nucleosomes, not between every 2, but after every 4 nucleosomes, something like that. Then we decided to make temperature melting of the chromatin of the mutants and wild type. As you can see, the melting of wild type is showing 3 peaks, when we can see only 2 peaks in the mutant without linker histone. And finally, we could even found in IFM images a well-built 30 nanometer fiber. But we cannot find such a fiber in mutants without H1. This is the first evidence that there is a 30 nanometer fiber in yeast, and the evidence that it disappears after removing of H1. So, from this sort of experiments, we think that H1 is very heavily involved in building of chromatin, higher order chromatin organization in yeast and probably in other organisms. It is easy to work with yeast because they've got only 1 gene, since in humans the linker histone genes are 11, so we've got 11 different variants of H1, and it is very hard to delete all of them. Okay, so let me go back to my previous experiments. Here I expressed long time ago a linker histone from sea urchin. Other people expressed in yeast different H1 histones, and they couldn't obtain the same result. One laboratory expressed the same histone in the yeast, and they obtained the same results. So it is proven by others, not by us only. But other histones couldn't make apoptosis in the cell. I mean, H1 histones. So the reason for that is because we've got different variants of the histone, and probably they've got a little bit different function in the chromatin. And because of this, we went to one of the most interesting fraction or variant of linker histones in human. It's called H10. And we tried to see, it is important, well, it is known that it is appearing during differentiation of the cells. So it is also called H10 is differentiating dependent histone. We took samples from glioblastoma multiforme tumors, which is a brain tumor, incurable, and wanted to see what is happening with this differentiate dependent histone. As you can see, there are different grades of the disease, and different grades possess different percentage of this histone, which is the percentage, 100% is the whole H1. And these different percentages are showing the quantity of H10. So we think that this histone is somehow involved in the differentiation of the cells. This is known already, but in these severe tumors, it has to be removed. It has to be removed in order cells to go without differentiation and make big tumors. So what are the conclusions from our work until now? Linker histones exist in almost all eukaryotic cells. Linker histones are among the major players which are building high-order chromatin structuring. And they've got different variants, and probably different variants are doing different things in the chromatin organization, and of course, they're making different things about transcription and controlling fine-tuning of the transcription and probably other processes which are happening in the chromatin media. Well, I've got 5 more minutes, so I can take them for questions. But first of all, let me show the people who are involved in this work. I forgot them because I'm first here. And I show myself here because I started the work and I worked a lot with my hands on it. But people like Milena Georgieva, who is associate professor in my lab, and she was very deeply involved in the work, and tomorrow she's got a lecture here, a talk here about linker histones and aging. And then another one professor, which is Desislava Staneva, who helped me a lot. But of course, people from my lab, like PhD students and graduate students, are helping me with mostly with their behavior and their helpful intentions. Thank you.
Questions?
Thank you. I was just wondering, on the yeast, were those knockouts? Were those H1 knockouts and you expressed exogenous sea urchin H1, or did you overexpress H1?
No, because it was done a long time ago, then we couldn't knock out the gene, so expression of exogenous was over the histone which yeast anyway possessed.
Okay, very nice talk, George. At certain moment you mentioned that by comet assay you are able to recognize the type of cell death and you are able to discriminate between apoptosis and necrosis. Could you tell me how?
Well, it is because of way of cuttings, you know, because in apoptosis you've got nucleases which are making usually they are making double-stranded DNA, and they even cut DNA, digest DNA to nucleosomal leather. So nucleosomal leather is usually a marker of apoptosis, as you know. So by comet, you can see double-stranded damages. It is another shape. This is recognized by the program, by the software program. So in necrosis, it's not the same. The profile, I mean the shape, it's comet shape, it's called, and it is freely available. I mean, it's freely available in our app, and we published it. So sure, but I will want a coffee.
Is there pro-apoptotic signaling between yeast cells, or does apoptosis occur simultaneously in only a single cell?
Okay, as I know for now, usually they are dying alone by apoptosis. However, you cannot exclude some very, very close contacts between signaling between yeast, which are when we talk about colonies, yeast colonies. But I'm not sure you can measure signaling, apoptotic signaling between yeast in culture, I mean in liquid culture. So I can talk a lot about the colonies and their signaling, but it is not place for now.
Hi. So you said that there are different variants of histone H1 in yeast compared to humans. And I was wondering if perhaps you had any insights into how these different variants could affect the three-dimensional structure of chromatin in yeast versus in higher order animals such as humans. Yeah.
Right in the target. Okay. Your question is right in the target. We did it. We expressed H1 which is very important, I think, in yeast. And we are now in experiments to see how this particular variant is changing chromatin, I'm sure. Usually people are looking only on the nucleosome level and they say, well, nothing changed. But we, as you saw probably, we look at deeper in the detail, and usually we are seeing small differences. So I expect difference in the chromatin organization, not so big. If you ask me about the phenotype, actually, by microscopic observation, nothing changes in this. They didn't become cubic, or they didn't start to go in the dark. So the phenotype is not changed by looking on that. You have to go deeply, and there are experiments with, but in human cells, by deletion one by one these genes, and actually they couldn't find the phenotype. But if you delete all of the types, the laboratory of Arta Scaucci did that, the cells are not dying, but they could not make, you cannot make multicellular organism. So they tried to do this in mouse, it couldn't be done. So probably these linker histones are important not so much for the unicellular organism, they're important for the multicellular organism. How? I don't know.
So you still expect it to have the overall fractal globule structure at the macro scale?
Well, it's a good question, and therefore I checked exactly this title for talk. Here, as you see in the picture, a lot of people just keep the H1 from the general picture. And here, even in this conference, only two of the speakers said a little bit about linker histones. So when you make a different kind of models of chromatin, you should involve H1. It's there, it's abundant, so it is causing some changes.
And so for time, let's thank the speaker.
Thank you.